Um, so good morning, everyone. Good afternoon, everyone. And thank you for joining us today. So today we have another yellow webinar. And it is my pleasure to introduce our guest speaker of today. This is Professor Sanda Win, who is joining us from USC, I think I will pronounce it correctly, USC Research Center for Liver Disease at Keck School of Medicine of University of Southern California. She is assistant professor there, and her major research interests are in the field of steatohepatitis, so alcoholic, non-alcoholic one, and also identification of therapeutic targets. Also drug-induced hepatotoxicity, mitochondrial bioenergetic signaling and in liver disease and oxidative stress. And her current work focuses on non-alcoholic fatty liver disease, metabolic stress and mitochondrial dysfunction, as well as sex differences in acute and chronic liver diseases. I hope I got all of that correctly. So today, Professor Wing will be sharing insights on how hepatic mitochondrial SAB level modulates mitochondrial stress, steatohepatitis, metabolic syndrome, and hepatic fibrosis. Yeah, before we start, I would just like to remind you, everyone, that if you have any questions during the presentations, you can just post them in the chat. And then after the presentation, at the end, we will have some time for a short Q&A session. And with that, I would like to hand it over to Professor Win. The screen is yours, and you can begin whenever you're ready.
Hello, good morning, good afternoon to you, and good morning to Los Angeles. Thank you for the introduction. Today's presentation is the role of hepatic mitochondrial SAB level in modulation of mitochondrial stress, steatohepatitis, metabolic syndrome, and hepatic fibrosis. First, I would like to explain what is SAB. SAB is an mitochondrial outer membrane protein. SAB and phospho-GNK co-IP from mitochondria expressed after toxic stress. Adenoviral SAB protects against acetaminophen and TNF-galactosamine toxicity. This experiment was done to know the topology of SAB using proteinase K to digest mitochondria and block with N-terminus and C-terminus recognized SAB antibodies. Then we can identify the SAB topology. This is the topology of SAB in mitochondria, showing SAB C-terminus is facing cytoplasm and it has GNK binding site. And terminus in the intramembrane space. Human and mouse SAB protein sequence is similar, 95%. GNK is one of the important molecules in pathophysiology. Stress from the extrinsic or intrinsic stimuli activate upstream MAP kinases, MLK3 and ASK1, then downstream MAP kinases, MKK4 and 7 are activated. GNK is activated, which can cause responses such as adaptive response, defense and proliferation of the cell, or inflammation, insulin resistant, or cell death. Response determined by duration and extent of GNK activation, crosstalk, and content. Toxic inserts such as TNF-galactosamine, acetaminophen, ER stress, and lipotoxicity cause sustained GNK activation. Activated GNK translocate to the mitochondria. Next, my question is the GNK effect on mitochondria. Because of SAB is the GNK binding mitochondrial protein, we develop liver-specific subknockout mice and acetaminophen-induced hepatotoxicity. Acetaminophen lethal dose cause sustained GNK activation in cytoplasm and translocation to the mitochondria. In contrast, subknockout mice prevent sustained GNK activation in cytoplasm and translocation to mitochondria. Similar finding was seen in the TNF galactosamine model. To evaluate GNK direct effect on mitochondria, mitochondria was incubated with activated GNK, 1 alone or 2 alone with ATP, or inactive GNK 1 or 2 with ATP. Activated GNK 1 alone or 2 alone with ATP decreased mitochondria, oxygen consumption rate, and respiration. Furthermore, the inhibition of mitochondrial respiration and decreased oxygen consumption rate are GNK dose dependent in both GNK1 and GNK2. Next, GNK effect on subknockout mice mitochondria was examined. Subknockout mice mitochondria was prevent the GNK-induced inhibition of mitochondrial respiration and decreased oxygen consumption rate. Then, to confirm the KIM1 peptide effect on GNK and SAB interaction. KIM1 peptide is the peptide that prevent GNK binding to SAB. KIM1 peptide prevent the GNK inhibition of mitochondrial respiration and decrease oxygen consumption rate. In addition, KIM1 peptide prevent increased release of mitochondrial reactive oxygen species. Therefore, GNK-mediated inhibition of oxygen consumption rate and increased release of mitochondrial reactive oxygen species is SAB-dependent. Therefore, we summarize that GNK-dependent toxicities such as acetaminophen, TNF galactosamine, ER stress, and lipotoxicity activate the GNK. Activated GNK translocate to the mitochondria, bind and phosphorate SAB. This GNK and SAB interaction inhibit electrotransport chain inside the mitochondria. It causes increased release of ROS from the mitochondria, activate upstream MAP kinases, MAP3 kinase, MAP2 kinase, then downstream GNK is activated. Then vicious cycle occurs, and this event causes sustained GNK activation in cytoplasm. To examine, GNK and SAB are indispensable for liver injury. Liver-specific SAB knockout mice and GNK knockout mice were given lethal dose of acetaminophen. SAB knockout mice protects against the acetaminophen-induced liver injury. And GNK1 and 2 knockout mice also protects against the acetaminophen-induced liver injury. SAB knockout did not change the total GNK expression, as well as GNK1 and 2 knockout did not change the SAB expression. Therefore, both GNK and SAB are indispensable for liver injury. To compare global deletion of GNK1 and 2 and SAB, GNK1 and 2 are embryonic lethal, but sub-embryonic knockout has normal development and no phenotype change. In liver-specific, embryonic deletion of GNK1 and 2 knockout cause age-dependent titular hyperplasia and cholangiocarcinoma, but sub-knockout has normal phenotype. Next, liver-specific subdeletion effect on other liver injury model were examined. Liver-specific subknockout mice protects against CON-A, CHO2, and furosemide-induced liver injury. To identify the KIM2 as a GNK binding site on SAB, we express KIM2 mutant SAB in subknockout mice. Wild-type SAB were re-expressed in the subknockout mice, and KIM2 mutant SAB were re-expressed in the subknockout mice, then given the lethal dose of acetaminophen. As seen in the histology, KIM2 mutant mice prevent the acetaminophen-induced liver injury and GNK translocation to the mitochondria. However, sub-wild type expressed liver restore the liver injury. Next, we predicted that GNK phosphorylation site is serine 379, serine 424 on the sub. We re-express mutant serine 379 and serine 424 into the subknockout mice. Mutant serine 379 re-expression did not protect acetaminophen-induced liver injury, but serine 424 mutant SAB protects from the liver injury. Therefore, we identified that KIM2 is a GNK binding site on SAB, and phosphorylation site is serine 424. Next, to explore the effect of GNK phosphorylation of SAB in mitochondria. Active form of phospho-SAB is required to maintain the electrotransport chain inside mitochondria. Active phospho-SAB decrease after GNK translocate to the mitochondria. But subknockout mitochondria maintain active phospho-SAB inside mitochondria. Next, to examine the effect of decreased SAB activities on the mitochondria. Mitochondrial respiration using active phospho-GNK with ATP, PP2, selective SAB inhibitor, SAB inhibitor 1. SAB inhibitors inhibit mitochondrial respiration, same as the effect of active GNK. After acetaminophen toxicity, tyrosine phosphatase SHP1 was activated in control mice group. However, no activation was seen in subknockout mice. Tyrosine phosphatase inhibitor can prevent GNK-induced inhibition of mitochondrial respiration. This figure explains the GNK SAB-ROS activation loop effect on intramitochondrial signal transduction pathway. Activated GNK translocate to the mitochondria and phosphorate SAB. That interaction causes releasing of PTP and 6, also known as SHP1, which is a tyrosine phosphatase, and its dephosphorate active phospho-SAB. The active phospho-SAB inside the mitochondria is required to maintain the intramitochondrial electrotransport chain. So after releasing of PTPN6, dephosphorate the SAB to the inactive SAB accompanied with DOG4. DOG4 is the intermitochondrial membrane protein. So decrease inactive SAB can cause decreased electrotransport chain, and increased release of ROS from the mitochondria. This consequently leads to increased sustained GNK activation, decreased mitochondrial function, mitochondrial permeable pore formation, increased pro-apoptotic and decreased anti-apoptotic B-cell proteins. And this is interesting to note that sub-expression is very low expression in the young female mice. We discovered that low expression of SAP also seen in the postmenopausal woman. However, postmenopausal woman SAP expression is similar to men. Low SAP expression in female mice prevent GNK inhibition of mitochondrial respiration and release of ROS. To evaluate the importance of SAP level, we express SAP in SAP knockout mice. The bar graph shows the expression level of SAP in knockout mice. SAP knockout mice prevent APAP-induced liver injury. After re-expression of SAP, restore the liver injury. After re-expression of SAP, restore the injury after toxic dose of acetaminophen. The injury and GNK activation are dependent on SAP level expression. Therefore, level of SAP expression determine the extent of GNK activation and hepatotoxicity in response to APAP. Here is the regulation of SAP expression in male and female. In young female mice has high estrogen receptor alpha that regulate to increase P53 and then increase microRNA 34A5B. MicroRNA 34A inhibits SAP expression on mitochondria. In contrast, male mice has low estrogen receptor alpha and P53, then decrease microRNA 34A expression, and low microRNA 34A cannot inhibit the SAP expression. With this finding, I conclude that SAP is a mitochondrial outer membrane protein that transduces the effect of activated GNK to signaling pathway inside mitochondria to promote ROS production. The stress-responsive GNK mitochondrial SAP ROS loop sustained GNK activation is a pivotal in cell death in many models of acute liver injury. The level of SAP expression is a key determinant of susceptibility to and severity of GNK-dependent SAP liver injury. In addition, SAP expression is increased in the model of NOFLD, such as high-fat, high-carbohydrate diet, Western diet, and high-fat, high-cholesterol diet, and sub-andescent prevent the diet-induced NOFLD. The role of GNK is also important in NOFLD. The hepatic MAP kinase pathway leading to sustained GNK activation is an important mediator of pathologic and metabolic response to overfeeding. Hepatocyte-specific genetic deletion of GNK1 and 2 or MAP3 kinase, ASK1, MLK2 and 3 prevent diet-induced NOFLD. Then, what is the role of the JNK sub-ROS activation loop in NAFLD? Sub-expression is increased after high-fat diet feeding, and expression is dependent on duration of feeding. JNK activation is increased that was parallel with sub-expression and duration of feeding. The findings were similar in human NASH liver. Sub-RNA was increased in human NASH liver in RNA-scope study and microarray study. Sub-RNA was increased in advanced fibrosis of human liver. Therefore, to evaluate the deletion of sub effect in NAFLD, high-fat, high-carbohydrate diet fat control mice increase the fat accumulation in the liver, increased liver triglyceride, and increased serum ALT. However, high-fat, high-carbohydrate diet fat knockout mice protect from the accumulation of fat in the liver fibrosis, and increase the liver triglyceride level and serum ALT. JNK activation was seen in the control mice, but no JNK activation was seen in the knockout mice after high-fat, high-carbohydrate diet feeding. Next, hepatocyte-targeted GANET sub-antigen were given to the mice in last 10 weeks of diet feeding. Here is the experiment schedule. 8 weeks old mice started to feed with high-fat diet. Total feeding duration is 52 weeks. At the time of 42 weeks of feeding, GANET sub-antigen or control antigen were given to the mice. And total injection duration is the last 10 weeks of feeding. Diet-induced increased sub-expression, JNK activation, and ATF2 activation were prevent and reversed to the normal level of expression were seen in sub-antigen mice group. In H&E staining, sub-antigen injected group were decreased number of blooming hepatocytes. Fat accumulation were also reversed to the normal in viral staining. Inflammation cell infiltration was decreased in sub-antigen liver. Moreover, serum ALT, liver triglyceride, insulin resistant, and are also reversed to the normal level, but also protein oxidation and NASH score become decreased. Therefore, liver-targeted GANET sub-antigen reverse the established NASH to normal. In addition, sub-antigen reverse the established fibrotic liver to normal liver. Sub-expression is increased in NAFLD and NASH. Therefore, sub-transcription regulation is important to determine. AP1 binding site is present in subpromoter region. ATF2 binding area and joint binding area are present in AP1. Therefore, high-fat, high-carbohydrate diet fat, ATF2 knockdown mice were examined. High-fat, high-carbohydrate fat, ATF2 knockdown mice have low sub-expression and decreased JNK activation. Next, high-fat, high-carbohydrate diet fat joint knockdown mice were examined. Joint knockdown mice have a decreased sub-expression and decreased JNK activation. Furthermore, knockdown mice has a decreased SAB expression. And moreover, knockdown mice prevent the diet-induced excessive fat accumulation in the liver. Therefore, induction of SAB in NAFLD is regulated by mitochondrial stress loop through sustained GNK activation and activation of phosphogene and phospho-ATF2 at AP1 response element. Another sub-induction is through ER stress at UPR response element at sub-promoter region, and this work is currently in progress. Therefore, I conclude that the stress-responsive GNK mitochondria SAB-ROS activation loop causes sustained GNK activation and is pivotal in cell death and metabolic impairment in acute or chronic liver injury. The level of SAB expression is a key determinant of susceptibility to and severity of liver injury by functioning as a rheostat which sets the level of sustained GNK activation according to the severity and duration of toxic or metabolic stress. SAB-induction in NAFLD is mediated by interplay between SAB-amplifying AP1 activation loop driven by the GNK mitochondrial SAB-ROS MAP kinase cascade. With this summary, I will conclude my talk. Thank you.